electrical engineers at the University of California, San Diego developed a technology that improves the resolution of an ordinary light microscope so that it can be used to directly observe finer structures and details in living cells. The technology turns a conventional light microscope into what is called a super resolution microscope. It involves a specially engineered material that shortens the wavelength of light as it illuminates the sample. This shrunken light is what essentially enables the microscope to image in high resolution. The researcher says that this material converts low resolution light to high resolution light. It is very simple and easy to use. Just place a sample on the material, then put the whole thing under a normal microscope. No fancy modification needed. The work, which was published in the journal Nature Communications, overcomes a big limitation of conventional light microscopes, that is, low resolution. Light microscopes are useful for imaging live cells, but they cannot be used to see anything smaller. Conventional light microscopes have a resolution limit of 200 nanometers, meaning that any objects closer than this distance will not be observed as separate objects. And while there are more powerful tools out there, such as electron microscopes, which have the resolution to see subcellular structures, they cannot be used to image living cells because the samples need to be placed inside a vacuum chamber. The major challenge is finding one technology that has very high resolution and is also safe for live cells. The technology that the research team developed combines both features. With it, a conventional light microscope can be used to image live subcellular structures with a resolution of up to 40 nanometers. The technology consists of a microscope slide that is coated with a type of light stringing material called a hyperbolic metamaterial. It is made up of nanometers thin alternating layers of silver and silica glass. As light passes through, its wavelengths shorten and scatter to generate a series of random high-resolution speckled patterns. When a sample is mounted on the slide, it gets illuminated in different ways by this series of speckled light patterns. This creates a series of low-resolution images which are all captured and then pieced together by a reconstruction algorithm to produce a high-resolution image. The researchers tested their technology with a commercial inverted microscope. They were able to image fine features such as actin filaments in fluorescently labeled COS7 cells, features that are not clearly discernible using just the microscope itself. The technology also enabled the researchers to clearly distinguish tiny fluorescent beads and quantum dots that were spaced 40 to 80 nanometers apart. The super resolution technology has great potential for high-speed operation, the researchers said. Their goal is to incorporate high-speed, super resolution and low phototoxicity in one system for live cell imaging. The research team is now expanding the technology to do high-resolution imaging in three-dimensional space. This current paper shows that the technology can produce high-resolution images in a two-dimensional plane. The team previously published a paper showing that this technology is also capable of imaging with ultra-high axial resolution, about 2 nanometers. They are now working on combining the two together.